It's my pleasure to introduce my longtime friend and colleague, ISB Professor Sui Huang. Sui is a molecular and cell biologist, but he's very unique in that he has a very strong background in theoretical biology. He has devoted his research to understanding the very phenomenon of cancer, and he does that from a complex systems perspective. So, cancer is a really complicated disease that can emerge because of many, many reasons. And there's been a central paradigm of how there's a genetic foundation to cancers that Sui has challenged. And this is what we'll hear about. It's going to be an exciting presentation. So before coming to ISB in 2011, Sui held faculty positions at the of Biocomplexity and Informatics at the University of Calgary, and teaching at Harvard Medical School's Children's Hospital. He grew up in Geneva and Zurich. He received an MD from the University of Zurich, and soon after that, he was the first recipient of the PhD program for Physicians Award from the Swiss National Science Foundation. And then he earned his PhD in molecular biology and physical chemistry. So since he's come to ISB, Sui has continued to work on his central theme of uniting experiment and theory to understand fundamental insights into the essence of what makes us multicellular and what causes cancer. And just 2 months ago, Sui and his colleagues published a really interesting essay in PLOS Biology. And the title of the essay was The End of the Genetic Paradigm of Cancer. It's a really provocative topic, something I've followed for many years. And I think this was a fabulous read. If you get a chance, I highly encourage you to read Sui's essay. And so, but you'll get to hear from him directly today. So, Sui, it's my pleasure to give the floor to you.
Thanks very much, Nitin, for the very kind introduction and for making it very easy for me to attack this very complex topic. Today, I really would like to be more positive and not talk about the cracks in the cancer paradigm, because that would require us to explain the exact nature of science and what is, a shortcoming. But I like to go beyond that and give you a view beyond the so-called genetic paradigm of cancer, because that would open a new vista on, understanding why so many treatments fail and what we can do about that. And I like to start with thanking a lot of people that really inspired me to think far beyond what is normally known. So I was very fortunate to have these giants of science on whose shoulders I could stand and who taught me how and encouraged me how to think beyond existing thinking, especially for this talk now, Judah Folkman at Harvard Medical School, a cancer researcher, and Stuart Kaufman, a theoretician in complex systems. And then, of course, many students and postdocs and collaborators who over the years have provided so much stimulating data and insights and discussions that help shape this view that I would like to present you today. So it's about going beyond a paradigm, and that's very abstract thing. So let me start in a concrete manner with this quiz here. Look, the picture on the left, what do you see? And what's wrong with this guy? That's a multiple choice question. Choose a statement that most logically can explain this behavior. I know we don't have feedback at this point, but you can try it on your own and think which answer is the best. There's one good correct answer, and that's most people would choose C, right? Because guarantees that you are logically correct. Either you cannot read or you cannot count, and that makes you do this behavior. But if you look at the numbers, it's actually different. And I would propose that another answer is he can read and can count, because that's what most people do, but he's a jerk. So it's a totally different type of explanation. And it becomes clear if you look at the numbers. It's very unlikely that somebody cannot read or cannot count, or even less likely both. So the quantitative details that underlie the premises of your logics might be so small, the numbers, such that they're just improbable. And so that means that sometimes the solution lies outside of your conceptual framework of reasoning, and you have to consider context and, like here, personalities and many other things that were not part of your existing paradigm. So now, what's the problem with cancer research? It's a long story. As you know, we started the war on cancer 50 years ago with this bill from Richard Nixon. And then there was, for a brief moment, a glimmer of hope around 2000 when people invented the so-called targeted therapy, so very precisely targeting molecules that target particular molecular deficits in the cancer or undesired activities. But that didn't last very long, as you will see. And then this was followed by other initiatives. As you know, the Cancer Moonshot program, now in its second version, and the latest is the initiative by the newly founded ARPA-H agency that wants to transform cancer care. So these are the collective efforts. But the problem is that as we moved from traditional chemotherapy to these targeted drugs, it was soon realized that the gain of benefit was not that great. So here you see a summary and a couple of those in the literature where somebody measured for many drugs approved in this year here, how much do they extend life. So this is oral survival. As you can see, typically a new drug, these fancy drugs that cost several $100,000 a year, extend life by 6 months or less, which is already pretty good. So people start to ask, why is it that all these new drugs cannot really eradicate cancer? And so that led me to think about the genetic paradigm, which is the reigning paradigm of the last 50 years, I would say. And at the center of it are mutations. As you know, mutations are changes in the genome. So here I show some normal cells that are green, and then a chromosome, and a mutation will be indicated by this red spot, and that turns the cell to a red cell, which is a malignant cell. That's the core idea. And now, how did that influence therapy and promote this so-called targeted therapy? So once a cell is mutated, the mutation might, by chance, confer a advantage of proliferation. Cells becomes much more resilient and faster in the dividing, and that leads to a clonal expansion of the cells that have that mutation indicated here in red. And so that gives rise to the notion of a cancer cell, which is defined by a mutation. And if you look at the mutation more closer, we can now sequence them, and mutations lead to the activation of many proteins here in the cell that promote cell division, cell growth, or other malignant features. So these are the mutated proteins. And then it became clear that the goal would be to molecularly target them using a drug that is precisely tailored to take down one of these activated cell proliferation promoting proteins. So that was the idea of targeted therapy. I list here a few early ones, HER2, EGFR, and BCR-ABL proteins that are targeted by these drugs here that you have probably heard of. And since we can sequence the genome of the tumor, it's easy then to predict which drugs could target which type of tumors, and this is give rise to precision oncology and companion diagnostics, where you sequence the genome of a patient. Now, these tumor cells, they can continue the process of acquiring more mutations, and that leads to the somatic evolution of malignant cells, because you have cycles of mutation and then selection for the most actively proliferating cells. So that's the idea of the genetic paradigm. that comes from biology. And just to remind you of high school biology, that's essentially Darwinian thinking for cells in the human body. So in Darwinian evolution, let's take these moths here. They're white. Occasionally, you have a mutation and one become black. And that is okay, but it can become an advantage if this mutant sees a change in environment. For example, in England, when you had industrialization, the tree barks became black and dark, and so the darker moths would have an advantage because they are camouflaged and so much harder to be picked up by birds. And that led to a selection of the black moths. So that's a basic idea, the school book, textbook idea of Darwinian thinking, and people have just applied that to cancer and its evolution towards more and more adapted cells that can proliferate and take over. And that's the idea of clonal evolution of tumor cell populations, popularized by Peter Nowell in 1976. And not much has actually changed. As I just told you, that's the basis for all these modern drugs. So then came a lot of sequencing, because we do now can do a DNA sequencing deeply and at single cell level, and that exposed some cracks in the paradigm. For example, we have a extreme diversity and heterogeneity of mutations that really do not explain why a cell is more fitter and can be selected for. We do see some signatures for change that are compatible with the idea of somatic evolution, but very little quantitative evidence that some mutations actually confer a advantage so that the cell is selected for, and because of that, expanding. So most of what we see is neutral drift. We see just a cacophony of mutations. So something else is going on. And what is that? Now, if you look back at all these drugs, and you can ask a very fundamental question: what is common to all cancer treatments? And simple answer, they all seek to kill tumor cells. But there's another answer, and that is they actually do not work well, all of them. And so this raises a question whether could killing actually be the problem, because that's the commonality of all these drugs, right? Along with the fact that we really cannot eradicate cancer, except for, of course, surgery in early stage is still the only really truly curative therapy. So on the war on cancer, that is a DALI 3 or AI-generated picture. We try just to have more and more powerful, more precise weapons to kill very specific tumors. And now we find all these mutations that we can target directly with these new smart drugs. Whether they will work or not, I'm not so sure. That's why I wrote this article here in Medium, if you want to see. The point is that we should reconsider the actual killing process of a cancer cell. And that's actually not new. A lot of people outside of medical research have realized that. Here's General Templer from Great Britain. He said, I think it was the Indochina War, the answer to the uprising lies not in pulling more troops into the jungle, but in the hearts and minds of the people. You've all heard understanding the hearts and minds. Another person, more recent, Don Rumsfeld, said that on the war on terrorism after 9/11, today we lack the metrics to know if we are winning or losing the global war on terror. Are we capturing, killing, deterring, and dissuading more terrorists every day than the madrasas and the radical clerics who are recruiting, training, and developing those against us? So you see the balance between two opposing rates of a process. And again, here, the numerical details matter. Similar spirit articulated by Barack Obama. So rather than offer false promise that we can eliminate terrorism by dropping more bombs, we have to take the long view of the terrorist threat, and we have to pursue a smart strategy that can be sustained. We have to fight terrorists in a way that does not create more terrorists. And that was the idea that he also said we should try to contain terrorism and not eradicate. But these kind of deeper philosophical ideas, they just don't penetrate into a cancer researcher because we can now just target mutations, right? So this pertains to the problem of backfiring. Every time we try to intervene with complex systems, the war on terrorism, you can empower recruitment and motivation and resolve, as we just saw from Rumsfeld's talk, quote, failed coup attempts can lead to emergence of even stronger leaders. Sanctions that don't work will lead to a strengthening of the grip of a dictator. We know in politics that if you attack your opponents, you actually can energize their base as opposed to weakening them. And the latest example I find very telling, if you try to restrict export of technology, it just stimulates innovation. Unless, of course, you can fully stifle a whole development. And we think that that happens also with cancer. When you try to kill them, and it's almost never possible to kill all the cells. The cancer always comes back in a more malignant form. That's just a fact of life, a sad fact of life. And the idea is that in complex adaptive systems, which all these systems are, human societies, governmental systems, and so on, an incomplete intervention will rather backfire. So there's no partial success, and you cannot come back later and finish off. So intervention is a double-edged sword. So how can we deal with this type of counterintuitive findings? So we have to reach beyond current, rather simplistic reasoning. Sometimes formal theories are needed, and mostly we need to think in a more encompassing category. So I'll show you just a few paradigm shifts to remind you what has happened in cancer. There are two types. One is when people say we shouldn't focus just on the cancer cell, but look beyond the cancer cell. And the other is what I will present to you later, is that cancer is not a disease of genes, but we need to go beyond genetics. So these are the two major paradigm shifts that have happened or is happening. Beyond the cancer cell lead to the idea of the tumor microenvironment, where these processes like angiogenesis, inflammation, and so on are taking place. And there's also the idea of tissue organization fields a more holistic approach by Soto and Sonnenschein. In the case of seeing beyond genetics, what we have, and I will discuss later, is what we call non-genetic causes of phenotype change and the plasticity of cells due to cell state transitions. So in the first case, that's very easy. People soon realize that the tumor is not just the cancer cell, or many of them that grow, but also many cells in its environment. If we abstract away, there are many cells in the tumor tissue, and many people have said that it takes a village to grow a tumor. So many support cells that contribute to tumor growth, and that's called the tumor microenvironment. Those cells talk to each other and form a very complex system that is very resilient. This idea also led to new openings for therapies. So we have several concepts here. We have the tumor-associated macrophages that are bad, they cause inflammation. We have cancer-associated fibroblasts that also support tumor growth. These are these normal connective tissue cells. We have immune cells that can support tumor growth or defend the body against tumors. And then, of course, we have tumor angiogenesis, the new blood vessels that grow into the tumor and help nurture the tumor. And that led to many new ideas. For example, we can block angiogenesis, we can stimulate the anti-tumor immunity to fight the tumor. That is now, of course, the best known and the latest and quite successful therapy. Just one example here, the anti-PD-1, or Keytruda, which I will come back later. And then people try to also block the immune defense that the tumor mounts to escape this anti-tumor immunity. And then all the new approaches try to suppress inflammation that contributes to tumor, or just directly kill these cancer-associated fibroblasts. So the whole idea is often referred to as stromal reprogramming, and the central idea here is that you are not killing the cancer cells directly. So that was beyond the cancer cell, and now beyond genetics. What do we have here? Again, this is this genetic paradigm, and if you look at the change in the genetic paradigm, you need a mutation to generate a new cell phenotype, as shown here. But cells are plastic. They can change in what is called a state transition from this green cell to the red cell without changing the genome. The one genome can have multiple phenotypes, and that has many ramifications for changing of cell types. And unlike mutations, which are rare and random and slow to show up because you require selection, these type of state transitions are frequent. They can be regulated by specific signals, and they really have immediate effect without much selection. So in the genetic paradigm, you need a mutation to explain the emergence of a new phenotype, whereas in the non-genetic plasticity case, you have cell state transitions, and you have one genotype that can generate many phenotypes. Also, it's important to know that these type of genetic transitions, non-genetic transitions, play a key role in not only in tumor development, but can also be harnessed for therapy. And another difference, mutations are the consequence of X-rays, mutagenic agents, and so on, hard to control, whereas these state transitions can be induced by non-mutagenic agents, of which there are many, and chronic cell stress and a lot of things. So that opens the door to explain cancer without mutations. And as you know, like microplastics, a lot of things, toxins do not actually cause mutations, hormones, and so on. So it's a new view that provides a new explanatory power. So let's look at the state transition. That's a natural biological process. It accounts for the fact that we have so many cell types in the human body, all generated by the same genome without mutations. And that's essentially the core of development of multicellular organism, because one same genome without mutations can generate thousands of cell phenotypes. So that's a new paradigm that I like to present to you, and it requires normally some math and abstraction, but I try to make it very simple with cartoons. So we are talking about state transitions without changing genes, going from one state to another. And I have to take a step back and talk to you about linear thinking, because we want to move into the territory of non-linear thinking. So typical linear thinking in a traditional view would be there's some cause, it activates genes through a chain, and then you have some effect. And sometimes the gene is mutated, it enhances that effect, for example, cell proliferation in the case of cancer, and that is what you want to target with a drug. So a drug essentially interferes with this chain of causation. And that reminds me of something, and I think that is not the correct way to view. Namely, you have these Rube Goldberg machines, where you have one switch in a chain of a lot of events that lead them to, in this case, to squeezing of toothpaste. That's not how the organism works, but this type of linear thinking has worked well for a very long time in classical physics. So what can we do beyond linear thinking? And that's nonlinear thinking and system thinking. And here is where the notion of state transitions comes in. So you have a state, A, and every state of a cell is defined by the expression level of many genes, of all the 20,000 genes in the genome. So if you go from one state to another, you have to change the gene expression profile of all the 20,000 genes. And as we imagine, that's very complicated, and it is highly regulated and constrained by the gene regulatory network, GRN. Many people at ISB working on that, including Nitin, has done a lot of work on that, and others. We just try to understand changes based on regulation of genes in this network. And we can also think of drugs not as inhibiting a step in this chain of causation, but they inhibit a transition of a state A to a state B, and without initially actually worrying about what is the exact gene target. So genes act in concert via a gene regulatory network, and thereby they establish cell states defined by gene expression profiles, and then they also control their state transitions. So that's the cell state transitions I was talking about. And just as an important detail, but it's really important because the arrows here that you see in these arrow schemes in many textbooks, that's a part of a causation. It's not a state transition, whether in this dynamical systems view, we have a true state transition of a complex system in one state to another. And now let's talk about influencing such state transitions. So we want to come up with an intervention. If we want to go from state A to state B, for example, changing the ideology of a society, or moving from terrorism to peace, or now in biology from cell state A to cell state B. These are highly complex systems that needs to be changed as a whole. And reality is just more complicated than just promoting this transition. And as shown here, what we often see in reality is that the trajectory splits, and that's what causes problem. So instead of going to B, it can also go to C. And these two states manifest an inherent dualism in the system of often two polar opposites as a potential outcomes of a perturbation. And the reason for that is that so-called nonlinear dynamics, and I won't have time to go into the details, suffice to know that a system cannot have inherently many outcomes, which by themselves are also distinct stable states. That's why it's called multi-stability. Then the second reason is a system is a composite of many subsystems. And so that some subsystems, in this case, the cells of a tumor, can respond in one way and others in another way. And that's why we get partial response. And a simple explanation, without going to the equations, would look like that. Why do we have this split if we want to go from A to B? Cell states are kind of these attractor states, these valleys in the landscape, very abstract mathematical construct, so-called similar to the potential landscape you know from physics. And going to another state requires a destabilization of the original state, but that makes you go through a state where you lose control, a instability, a transient instability, such that cells not only go to B, but because they're on a hilltop here with high potential energy and many outcomes, it can go to just the opposite direction. And so you get always the undesired outcome that is opposite to what you want. And one can see that with cells. So you want to kill cells to bring them to this apoptotic or cell death state. But many cells that don't respond that way will just go in the opposite direction and become stem-like cells that are much more resilient. And we can see this also in tumors. Then patients that don't respond as desired, they often exhibit a hyperprogression, to doing actually worse than non-treated patients. So the new paradigm now could be used to explain why we have recurrence of tumors in a much more malignant form. In the traditional view, you have a mutation, and you kill the normal cells, the mutated cell would survive for whatever random reason, because that mutation conferred some resistance, and then these cells would proliferate, and that's the selection I mentioned. And that's called the survival of the fittest, according to Darwin. In the new view, we know now, because of the heterogeneity of cells in a population, you never kill all the cells. Even at the high dose, as you can see here, many cells still survive just by pure chance, because every population has some distribution. And so those non-kill cells are perturbed. They are not just there, they underwent a near-death situation and are highly stressed. And because of that, they often move in the opposite direction. So if this is the goal, to move cells into the right-hand side, because you destabilize the whole system, some cells that are not killed, like on the right, they can spill over to the other side where you have a state. You know, every position is a state, is a phenotype that correspond to a more stem-like state, and these cells are more resilient and they can expand. So this is not survival of the fittest, because individual cells that are not killed, they actually change themselves through plasticity. And that's then we also find something very disturbing, and that is the dead cells in the tumor will further stimulate this transition to this more stem-like state. And that is essentially the principle of Friedrich Nietzsche, who said, what doesn't kill me makes me stronger. So every cell responds. If you are not killed, you actually emerge stronger. And that is exactly, of course, what many of us see in everyday life and in society, right? And the same thing happens with cancer cells. And that's the basis for why therapy can backfire. So I'll show you some data in mice that we have done with Deepak Panigrahi in Boston. Here is a tumor growth curve of a very aggressive tumor. It's called Lewis lung tumor. The black one is untreated. If you treat with standard chemotherapy, you can slow down growth, as seen here in this red curve. That's fine. That's a standard model that we use to evaluate new drugs. But now we create a new situation where we have very slow-growing tumors by just injecting many less small number of tumor cells, 10,000. When we create the tumor, you can see tiny tumors here in these shaved mice. And now the exact same dose of cisplatin, if you now treat these mice with the same dose, suddenly the tumor grows. So you are awakening a giant. And luckily, in most situations in the clinic, we have the situation here on the left where we actually suppress tumors, but we have to be careful, especially with chemo in small tumors. You can actually make them grow, and that's when the treatment can backfire. And you can see this in many tumor models. This is another mouse model, a breast cancer, and here we treat with anti-estrogen, fast-growing tumor. This blue line here is suppressed, as seen here in this dashed line. But if a very slow-growing tumor, now treat it with the same dose of the drug, you actually awaken the tumor. Here another example with the lymphoma case. So and we have seen this for many cancer types. It means the treatment is more complicated than just killing as many tumors as possible with maximally tolerated dose, that as is the traditional scheme. So you make tumor either weaker or die, or you make them stronger, and 3 is the ratio of these two responses that determines the net benefit to the patient. And so coming back to this idea, there's a theory behind the whole thing. I don't want to bore you with that, but just show you that these gene regulatory networks can be framed as a landscape, and that's an old idea. I have now a much more complicated landscape than the one I've shown you. Every cell would map onto this little valley, which is an attractor state, and a state transition is a path on this landscape, and you will seek normally, in normal development, the best path to get to another state. And this is the old idea of Conrad Waddington, who proposed as a metaphor this type of landscape that you might see at many places. And the core idea is that attractor state represents cell types. And we can use this now to explain the existence of cancer. I don't want to go into details. That's very much described in the paper that Nitin mentioned. So normally cells would go down and form normal cell types. down the trajectory of development. But in this landscape, mathematically, there are many regions with many attractors that are never occupied. So cancer is essentially when cells go into these unoccupied, unevolved attractors that are also stable, but because they never evolved, they don't know how to move back to a normal trajectory, and that is how then cancer can arise, and they accidentally go into these attractor states, these cancer attractors, due to a non-genetic perturbation or a genetic mutation. So this new concept does not exclude genetic mutation. It just tells you that with chronic push, you can also get to these cancer attractors without much of mutations. Yeah, as I mentioned, these cells cannot go back, or it's very hard. That's why differentiation therapy, to turn these cells into normal cells again, as many people have tried, is very hard. Implications for cancer treatment for the last 5 minutes. With this idea, we have the following concepts. One is that thou shalt not kill, or only kill gently, and then you should clean up the dead bodies because the dead cells contribute to inflammation that promotes tumor. Always consider the broader context, not just the cancer cells. The entire tissue is a village. And then we should take care of not stressing the cells that are treated because they can become more malignant. That is the bifurcation that I've shown you. And so we have been looking for substances, drugs that do that. And this goes back now almost 10 years. You can see here traditional killing of tumors with chemo. But what is new is immune checkpoint inhibitors, which seeks to stimulate immune cells to attack the tumor, which expresses these tumor antigens, the red triangles. And that's a goal, and it is by activating the adaptive immune system. But for this to work, and that's known in immunology, the tumor antigens have to be presented first. You have to immunize your immune system. And that's done in this process here. When cells die, they get eaten up by these macrophages. Ideally, they would then take the tumor proteins and present them to the immune system, so this complicated process of cross-presentation. But what often happens is that these macrophages just trigger inflammation, cytokine release, and also immune suppression because it thinks this is normal tissue and not a bacterial invader, and that leads to more tumor progression because you have more inflammation. So we're looking for a drug that inhibits that red arrow here, and that's called TP317 now. But it turns out that this drug also inhibits many other, also promotes what you want. It promotes antigen presentation. So we found that very neat. And it turned out that this drug also promotes antigen presentation in the tumors, which is also needed for the T cells here to recognize a tumor. And so with that, we can turn an immune cold tumors to an immune hot tumor, which is the term people use when a tumor has antigens presented that make it susceptible to immune checkpoint inhibitors. And that drug is essentially a stabilized form of a endogenous mediator, resolving 1, and it's being made thanks to the vision of the CEO and co-founder of TT, the company, and I disclose here my relationship with that. We have been working with this drug for many years, not knowing what it does, but now we know that it really doesn't kill the tumor cell, but reprograms the stroma such that it's just conducive to respond to immune checkpoint inhibitors, and Deepak Panigwai helped with a mouse experiment. So we have promising preclinical data, excellent safety and pharmacokinetics profile in phase 1, and we are now trying to move to phase 2. And just don't want to go into details, but drug really work well in mice. You can see in combination, TP317 in combination with this anti-PD-1 inhibits a lot of tumors that normally would not respond to the immune checkpoint inhibitors alone. For example, in this case here, the drug alone actually is pretty good, but together with checkpoint inhibitors, it really can suppress a tumor which normally is not responsive to these anti-tumor immune drugs. And finally, we also show now in details that I don't want to go into, that if you look at the gene expression profiles of these tumors treated with the drugs, they really increase all the genes that are needed to present the antigen to the immune system, and they inhibit, as you can see, the inflammation promoting genes that would actually accelerate tumor growth. So I stopped with this. I know it was a lot, and that's why I just want to summarize with just pictures. I started with this strange situation of something that really you need to explain only by thinking in a more encompassing category, as opposed to staying with your logics. And then what doesn't kill me can strengthen me. I hope you get that idea by now. So with more pictures to illustrate the idea, you cannot always come back to finish off. So a wounded animal can be more dangerous. And that's why treating tumors are difficult. It's similar to this multi-headed hydra that if you cut off a few heads, more will grow back. And I thank you for your attention, and we got to take questions. Back to Nitin.
Hey, Sui, that was fantastic. Always very inspiring to hear about this different perspective that most people don't necessarily understand or have thought about. Um, so there's a lot of questions in the uh, Q&A window, but let me start off by asking the theory you laid out for how cancer emerges and um, how does that influence our thinking of designing treatments as we go forward in terms of um, treatments that are therapeutic and are less likely to lead to resistance?
Yeah, I mean, it's a great question that we all ask, right? I only present a view that considers, the tumor microenvironment, which more and more people do. I know we still can try to push harder on the killing front, and of course, as you do, also be more, uh, specific with resistance, right? Because the response the tumor mount against your drug is resistance, and that's not just selection of pre-existing resistant cells, but they move into a more tolerant, or how to call it? You have a term for that, tolerant state.
Yeah.
And that's also an option. It's just hard. You know that, right? There's so many cells, so many possibilities.
Yeah,
But
but
that's
one of the things that building on your theory, one of the things that uh, my group and other groups have done, and you've been part of this as well, is to exploit this capability of tumor cells to essentially change phenotypically into new
yes,
states,
yes, yeah,
to predict what those states might look like. Because that,
yeah.
as you pointed out, and we've also shown that the tumor cells have a gene network that is operating, and as long as there's no selection, the same gene network can be used to predict how that tumor cell may go into a new state that is more tolerant and eventually gain resistance. So that allows for forward-looking treatment strategies with sequential treatment and so on, right?
Yes, exactly. And I think that's a good opportunity for single cell analysis. You need single cell analysis because if you perturb the tumor with standard drug, they move into new states. And now we can actually start to characterize those new states they move to. But it's, this is only the beginning. Very few people do that. Even, in the 1980s, we had drugs specifically preventing resistance and the resistance genes from coming up, and those didn't work well. So it might be very complicated. We have to prevent many transitions into multiple fates, not just one. And for me, it's like herding cats, they go in all directions and you have to really capture the most essential ones.
True, true. There are a bunch of questions that kind of hit the same theme of, this is a paradigm shift in how you think about cancer and how you design treatment strategies or find new treatment strategies. And so the question is, where does the cancer field, the oncologists and clinicians, where do they fall on the spectrum of buying into this paradigm, and how do you think that is influencing, if it has influenced their thinking about approaching cancer treatments in new ways?
Yeah, that's important to think because, ISB, we used to think always in the forefront of new ideas. But my impression is, I was just at the AACR, a lot of people think in terms of cancer mutations still. There's still sequencing companies coming up and using AI to understand the mutations. But the notion of plasticity definitely is spreading, as you know. Everybody talks about that now, but I'm not sure people talk about controlling plasticity so much yet.
That's true, yeah. And in this full disclosure, I also have a spin-out company in this space that is trying to build on some of this paradigm and bringing cancer treatments that are more linked to cell state rather than mutations. And there is, I think, evidence now that mutation-based precision medicine has really not met its promise or potential. And one of the reasons is exactly what you're pointing out is mutations capture a small sliver of the underlying biology of tumors. But that essentially means there are ways with our new technologies for profiling, RNA sequencing in particular, that you will be able to design treatments that are targeting cell states rather than mutations, correct?
Yeah, exactly. And as you know, mutations, people now literally talk about targeting mutations, but only at best 20% of cells have that mutation that you target. Sometimes just 1%. And so it's very dismal. And but a state, every cell has more or less the same state, so it's much more promising. It's just harder to translate into a drug.
Yeah, and here's in the context of what we just discussed, there's a important question because we still treat cancers in for a large part the same way that we did 5 decades ago.
Yeah,
Is you
still.
essentially kill, cut, burn, poison, right? Um, so do you think chemotherapy and radiation therapy will, slowly fade away into the horizon as less effective treatments because they probably are making some of these cancers more aggressive and worse?
Yeah, maybe they will be used in exceptional situations, right? A targeted therapy works in the rare case where you have a clonal tumor where the mutation has spread to every cell. This happens occasionally. They might work. And also with what worries me is with early diagnosis. Early diagnosis is good for surgery, but it's not good if you go with chemo after these small tumors. I mean, we see it again and again. I wouldn't do that.
That's true. I mean, a lot of these could be benign. I mean, they'll just,
Yeah, we
the
don't know.
body may probably resolve it on its own. So, in the context of this phenotypic landscape that you've described, there has been theory related to, metabolic theory, and there's, classic Warburg syndrome and so on. Um, those seem quite consistent with the theory that you're proposing. Is that correct?
Yeah, exactly. What we try to explain is why tumor revert back to ancient phenotypes. Ancient in the sense of, in terms of development, right? Embryonic cell, but also ancient in terms of evolution. This Warburg effect and anaerobic, oxidation, these are ancient programs that are deep in the network. And if you fail to differentiate to mature cells, you just reactivate them. So it kind of fits well. And also we look at data, we look at the age of oncogenes. Many people have done that. And that fits with the theory that tumor revert back both in terms of evolution as well as in terms of embryonic development.
Yeah, yeah. Um, I think there's a lot of questions about, the um, translational potential of this theory. How soon can we expect this to impact patients? And some people have personal, and all of us have been touched by cancers, and some people on this thread
Yeah.
have family members who have been affected, and I think they would like to know, what can they expect?
Well, we try hard to raise money, right, for phase 2 studies. We actually hope sometimes it's so non-toxic, our drug, that we hope to be able to do some compassionate use case, but it's tricky. I think what there will be a big wave of drugs based on the theory that you don't want to poison and kill the tumor cells that are inherently less toxic, and that's a good thing, right? And then there are many people trying to push using supplements and all these things, the so-called GRAS, G-R-A-S, generally recognized as safe. I don't know, some vitamins or things like that, and hoping that those compounds also have this cancer modulating activity as opposed to not killing them, because they are inherently not poisonous, not toxic. And so many people screen for those already widely used supplement, whether they have these activities to modulate something, activate the immune system, or cause cells to behave better, transitioning to a better state. But the most concrete advance, I think, maybe, I know, of course, bias is the TP317 that we're trying to develop.
Yeah, and just my a little perspective here is we have spun out a company out of ISP from my lab called Signomics that is trying to deliver some of these cell state-based therapies to patients um, now, and it's a startup company. I think the way the current field is operating now is you do have a few companies that are offering CAP/CLIA certified tests for profiling tumors. And
Yeah.
while traditionally these profilings have looked at mutations, more and more these companies are also providing RNA sequencing data. And as most of you know, and Sui mentioned, RNA sequencing captures the cell state. And using the kind of systems biology and network analysis we do at ISB, we can take that profiling data and elucidate this tumor cell state in patients and use that as a means for identifying which therapies are likely to be more effective. There is a complex regulatory landscape around this. It is hard to do this for cancers and patients where standard of care is already available. And I think as the field advances, I think initially patients who are at the most extreme risk of not responding to any treatments may likely see some of these leading edge treatments. But over time, as there's more evidence that this actually is a better approach than mutation-based approach, we think this will start then percolating towards.
Yeah, I think
More
too. I'm
patients.
actually hopeful, but one point thing that you mentioned about all these drugs you guys testing in phenotypic assays, the good point is really we require that the compound is not killing the cancer cells. That's, I think, a new idea should get into the back of our mind in developing drugs. You know, politicians, they know it by now, you know, the military strategies. You don't always want to just go with a hardest bombing of your enemy. So, but because these drugs are inherently not killing the tumor, they're much likely to be non-toxic. And as you said, I think in one of your projects, right, we can repurpose drugs known to be non-toxic or variants of drugs that are on the market, and that might accelerate a whole new field of drugs that are not intended to kill the tumor cell.
Yeah. No, I've
Yeah.
worked in this space long enough that I've come to realize that there's some additional paradigm shifts that are needed, especially in the
Yeah.
drug discovery domain, where pharmaceutical companies and the whole FDA regulatory process essentially requires that the single agents you discover, have killing effect on the cells themselves.
I know that exactly. These rigid paradigm affect regulatory, you
Correct.
know, structures, which also makes progress very difficult. Yeah. The other one is, for example, since FDA is so used to cancer drugs being toxic, that they don't accept a phase 1 in healthy people. You have to do it on cancer people, cancer patients that have no other options. And that also makes it progress very difficult.
That's true, and the numbers are too low to really learn anything from those. Yeah, but you know there is hope. I think we have better models, ex vivo models, where we can look at some of these drugs and drug combinations in new paradigm settings. And the evidence is coming now. I think there's, as Sui pointed out, I think there's general consensus developing that the mutation-based theory does not explain all of the complex resistance phenotypes we're observing. And so, out of necessity, I think more and more clinicians are looking outside the box. And I think this is an opportunity for us. And we have the tools and technologies and the theory that Sui has to kind of move in the new direction. Now, we just have a minute, Sui, so we'll have to close it here. And I just have to give some closing remarks. But just wanted to give you a huge thank you. And I'm sure people are clapping. We can't hear them. But this was an impressive presentation and really hopefully gave people this new dimension of thinking of what cancers truly are and how we should think about treating them. So thank you very much.
Thank you, Nitin, and thank you everybody for listening. I hope it was helpful.